This is one of the trillions of cells that make up my body. This material in the cell is called the cytoplasm. Over here is the cell nucleus. Inside the fragile nuclear envelope are the long, thin strands of DNA. As we've seen, DNA contains the genetic instructions that control the cell's metabolic functions and heredity. Sounds simple enough, but the process by which this genetic transfer occurs is sublime. It's the story of our next great discovery. Years before Watson and Crick had defined the structure of DNA, scientists knew that DNA was responsible for making proteins that resided in the cytoplasm. But they were puzzled by how the DNA managed to transfer its genetic information for making proteins through the nucleus wall. The answer to the puzzle came through the collective work of several different scientists. And like many discoveries, it began with an educated guess. The production of protein in a cell is seen here in red. Scientists found that cells with lots of protein production contained lots of this, RNA, a chemical similar to DNA, but with only one strand, not two. This led scientists to wonder if RNA was somehow involved in running the protein factories in the cytoplasm. To find out, once again, researchers turned to the bacteriophage. They detected that soon after the bacteriophage injected its DNA into a bacterium, traces of viral RNA appeared in the host cell, and the production of proteins began to increase. This was the moment of discovery. Here was a special kind of RNA never seen before. As its single strand enters one of the cell's protein factories, it communicates the messages for making new protein. Scientists called it messenger RNA. Today, many scientists believe that messenger RNA and other RNA molecules are descendants of the Earth's most primitive chemical materials, part of the primordial soup that cooked up the first living organism. As we've just seen, RNA translates instructions from DNA for making proteins. But what was the genetic code, the sequence of instructions that made this process possible? In 1961, molecular biologist Marshall Nirenberg and postdoctoral fellow Heinrich Mattei carried out a series of experiments to see if they could synthesize proteins in the laboratory. They already knew that there are 20 amino acids involved in the production of proteins. In their experiment, Nirenberg and Mattei were working with RNA. Like DNA, RNA is made of 4 chemical bases. In a eureka moment of discovery, they found that when 3 of the bases align in a specific sequence called a triplet, the triplet codes for a specific amino acid. The order of the triplets is the blueprint for the production of proteins. Today, Marshall Nirenberg is a research scientist at the National Institutes of Health. So what does this mean to me as a citizen, taxpayer, voter?
But what it meant is that RNA is transcribed from DNA. DNA is copied into RNA, and RNA contains the information that determines the sequence of amino acids in protein. So that what you inherit from your parents is the sequence of letters in DNA that determine how you synthesize, how you make all the different kinds of proteins that are needed for life.
That make me.
That make you, that make me, yeah.
Nirenberg and Mattei's discovery was the first crack in the genetic code, a peek at the secret vocabulary encrypted in each DNA molecule, enabling it to instruct the synthesis of proteins. Over the next 5 years, Nierenberg successfully deciphered the 64 triplets that comprise the entire vocabulary of DNA. The genetic code had been broken. And in 1968, the discovery earned Marshall Nierenberg a share of the Nobel Prize for Physiology and Medicine. What was this like? You make this discovery.
Well, it was opening a door to a toy shop. I mean, we could do virtually anything. We could decipher the genetic code. Every
living thing has the same code, right?
Yeah. There's one code that's used by every living thing on this planet. And it's the same, it's basically the same language that's used. And when we found that out, you know, I knew all about Darwin, I knew all about evolution, but this brought it home in such an immediate fashion. All living things on this planet are related. We're all derived from a common ancestor, and in our bodies all speak the same language. It had a very marked effect on me.
Where Watson and Crick identified the structure of DNA, Nierenberg and Mattei uncovered the blueprint for how the structure worked. The breakthroughs that led to some of the discoveries we've seen so far might never have happened if not for this familiar organism, bacteriophage. And our next discovery depended on it as well. For decades, it was believed that bacteria were completely vulnerable to invading bacteriophage. But in the 1950s, researchers found a wrinkle in that view. Certain types of bacteria were, in fact, resistant to being infected by a bacteriophage. How is that possible? The first answer was provided in 1962 by microbiologist Werner Arber. He found that some bacteria had enzymes that fought back against the virus by cutting its viral DNA into pieces. This restricted the virus from taking over the bacteria. So they were called restriction enzymes. But how exactly did they work? Among those hoping to unlock that secret was microbiologist Hamilton Smith. Working in his lab in 1972, Smith was growing bacteria and bacteriophage together when he noticed that the DNA of the virus was breaking down. Smith acted quickly. He purified the restriction enzyme, then he identified the exact place where the restriction enzyme had severed the DNA. Then came the moment of discovery. Smith found that the enzyme repeatedly cut the viral DNA in the same place. He had discovered the first site-specific restriction enzyme. With restriction enzymes, scientists now had a pair of molecular scissors they could use to cut DNA molecules, to virtually recreate nature. This is significant. Today, this ability to manipulate DNA is one of the basic tools in genetic engineering, what's called recombinant DNA research. Since Hamilton Smith's discovery, hundreds of restriction enzymes have been identified, and scientists are using recombinant DNA for a world of applications on which we depend. Everything from the creation of more effective, less expensive drugs, to the production of human insulin for the millions of diabetics worldwide who rely on getting their daily dose. With the discovery of messenger RNA, scientists had found the process by which DNA communicates instructions for making proteins in the cytoplasm. For decades, they believed this process operated by one simple rule. Instructions were coded from one gene in the DNA to one type of messenger RNA, which then produced one protein. But in the 1980s, this theory was challenged when scientists began detecting something new. Genes that coded multiple messenger RNAs, which then produced multiple proteins. It would take the combined efforts of several scientists to find out how this was possible. But finally, they had their answer. Through a process called alternative splicing, some genes are able to code for more than one protein. The discovery of alternative splicing was important because it gave scientists valuable new insight into the role that RNA plays in the production of proteins. And that insight helped researchers make advances in a whole range of biomedical applications. For example, scientists have used their knowledge of alternative splicing to create more effective painkillers. Some medicines are designed to block the production of specific alternatively spliced proteins and enzymes which regulate pain in the nervous system. London, 1985. A boy arrives by plane from the African nation of Ghana. The purpose of his visit? To be reunited with his mother. But custom inspectors are suspicious. The boy's passport appears to be a forgery, and there's no proof that the woman he's meeting is really his mother. The British government decides to deport the boy. Desperate, the woman turns to a detective for help. His name? Alec Jeffreys. Not a real detective, but a geneticist at Leicester University. Before we learn the rest of the story, a little background. By the 1980s, scientists had become aware that genetic variation among individuals is very common, and they had begun to link the presence of these so-called DNA variants to the presence of disease-causing genes in large families. For example, Huntington's disease. And that brings us back to Alec Jeffreys. In 1984, Jeffreys found something never seen before. It was a DNA variant formed by short, identical sequences of DNA that were repeated over and over. Jeffreys called these repeated sequences mini-satellite DNA. Then came the breakthrough discovery.
Well, what we're detecting are lots of bits of DNA, and in the humans at least, are clearly variable, very variable. And in fact, what we got was our first very, very murky DNA fingerprint. And at that point, the penny dropped.
Jeffreys' discovery made history when he was asked to help solve the mystery of the boy from Ghana. For the first time, DNA fingerprinting was put to the test. Jeffreys compared the DNA of the boy with the DNA of his supposed mother. The results showed a striking similarity in the mini-satellite DNA, proving beyond a doubt that the boy was her son.
So I was there when she was told, we've got the DNA evidence, it's been accepted, your boy's coming back, he's permanently with you. And it was the look in that lady's eyes was magic. And this was really, I mean, the first time DNA had ever done this. It was the first time that DNA, all this modern arcane science of molecular genetics, had actually been used to go out in a non-clinical context and actually directly help someone.